Guys, welcome to my discussion 6 concept review. Today we're going to be talking about gene regulation and how that affects protein expression and cell differentiation, and going over 2 main mechanisms of how gene regulation happens in prokaryotic cells. One interesting thing is that we only have 10 discussions total this semester, and we're already at discussion 6, so we're past the halfway point. So it's crazy how fast the spring term goes. But yeah, so getting on to the actual concepts. So we know that in a multicellular organism, all of the cells that are located in the organism contain the exact same genome. And so when we think about the different types of cells that we have in our human body, that can seem like a little weird because we look at the structure of a muscle cell, for example, and it looks super different than a nerve cell or a cancer cell or a blood cell or any of these types of cells. And so how can that be possible that they have such different structures and functions if they're all being based off of the same genetic code? And the answer to that is gene regulation. So essentially, even though every single cell has technically the potential to produce any proteins of anything in the body, the actual proteins that end up being expressed are going to depend on how the different genes are regulated. For example, we can think about our eyes. They contain rod and cone cells, which are photoreceptors, which are able to perceive light stimulus. But we can think, okay, example, with our muscle cell or our liver cell or something like that, there would be no reason for us to have photoreceptors. So it's more energetically favorable for the cell to say, you know what, since we don't need them, let's just not transcribe them, even though technically we have the instructions available to do it. So there's 2 main types of gene expression. There is constitutive expression, which is essentially a gene is always going to be on, so it's always going to be transcribed and make whatever protein it codes for. And so this is going to be common for vital processes for every single cell in an organism. So like if we think about all of these different types of cells, even though they are really different in lots of ways, there are still fundamental activities that they're all doing that are kind of like shared between all of these. So those like vital processes are going to be likely constitutively expressed because regardless of any specialized function, there are still some proteins that will be needed wherever. And then there's regulated expression, which is what we are really focusing on today. And so essentially this is the case where certain conditions or stimuli will tell us, okay, do we want the transcription of these proteins to occur or not occur based on like what situation we're in. So there's 2 different possibilities for how regulation can happen. And so in induction, the default state is that the gene expression is not going to occur unless we add a specific stimulus or condition that will activate it and say, hey, we actually want these proteins now, so let's turn this on and allow the reaction to occur. But conversely, we can also have repression, which is also called negative regulation, which is opposite of that, where we say, we're going to have this gene automatically be transcribed by default. So by default, the gene is on, unless we receive a certain stimulus or a condition, which is called a repressor, that causes the expression to now halt. So something is telling the cell, we don't actually need this anymore, so you can stop doing what you're doing. So I'm going to go over how this actually happens. So expression in prokaryotic cells is controlled by operons. And essentially, an operon just describes this set of things that ultimately control how genes are regulated in a cell. The operon refers to the promoter, the operator, and then genes, which are all controlled by the same promoter and operator. So the promoter itself is just a specific nucleotide sequence that lies at the very beginning of the operon that will bind RNA polymerase. So essentially its function is just to kind of recruit the RNA polymerase to the right spot and allow it to bind. And so that's kind of like the first step of allowing transcription to occur would be to get that RNA to the right place. The next thing, the operator, is kind of more like a gatekeeper of the RNA polymerase. So under normal conditions, when the operator is available and not mutated and functional, whatever, it's going to allow the RNA polymerase to move from the promoter to the operator and then down the DNA so that it can actually start transcribing. However, if there's something either mutated with the operator or if something is blocking it, if there's like a repressor attached to it, it will act as kind of like a physical gatekeeper to the RNA polymerase. So in this situation, we would have the RNA polymerase still bound to the promoter because the promoter is functional, but it can't physically reach those genes to transcribe because it's being blocked by something on the operator. And then lastly, I just want to talk about what we mean by these genes here. So these genes are, we call them structural genes in this case because they're going to code for things that are important for like biosynthesis and stuff like that. Any genes that are kind of linked together under this same promoter and operator are most likely going to be very similar in function. And that's because we would only want genes to be linked together when they kind of respond to conditions in a similar way. So if you think, for example, if we had genes for activation of a neuron cell versus a muscle cell, we would not want those linked together because the situations where we would want certain things to function in a neuron versus a muscle cell, the situations are going to be like pretty different from each other. So regulating them at the same time, like with one switch, is not going to be useful. But if you have something that like, but if you have proteins that are all super functionally related, it makes more sense from an energetic standpoint to just regulate them all at once. If they all respond to the same conditions in the same way, then why not just like link them together? So that's kind of why this has evolved. It's just easier for the cell. Okay, so first we can kind of predict what are some things that we could possibly do to the operon to alter the gene expression. And I kind of hinted at this a little bit, but going back here, we remember that the operon is a promoter, an operator, and the genes. So I already said we could modify this operator to block the RNA polymerase from transcribing. However, we could also block or mutate the promoter itself to prevent RNA polymerase binding to begin with. And since the promoter essentially is like recruiting the RNA polymerase, if it's not able to bind here, even if this operator is open, it's still not going to be able to transcribe because we're not even getting that RNA polymerase there in the first place. So those are just some examples of how regulation can happen. Okay, so there's two classic examples that are used in biology to kind of use as a demonstration of how gene regulation happens. The trip operon and the lac operon. And I wanted to start with the trip operon because it's a little bit simpler than the lac operon. But essentially, so the trip operon works in E. coli, and it synthesizes tryptophan whenever it's needed, but it's able to sense when tryptophan is already present. And if it's already present, then the cell has no need to synthesize the enzymes that are required for tryptophan to be synthesized. So it'll say, okay, we don't need this anymore, so we can stop this process from occurring since we already have the tryptophan that we need. So here, we can take a closer look at the actual physical structure. So here we can see the promoters here, the operators here, and then these are all the coding sequences that are kind of like linked together. So if you get this TRIP-E sequence being transcribed, all of these other ones are going to be transcribed. Like if transcription is happening, then it's happening for all of them. And this is useful because these are all the enzymes that are required for tryptophan to be synthesized. So instead of activating them all separately, we can just do them all at once. So these are called the structural genes. One other thing that I wanted to mention that I didn't earlier is that in some cases, the operator is technically part of the promoter, but I don't want you guys to get confused because even if the operator is part of the promoter, it's still going to be located at the very far tail end. So if we would just remember that the operator is acting as a physical gatekeeper between the binding region of the RNA polymerase to the genes, the operator is always going to need to be like sandwiched in between the binding site and the genes themselves because it's acting as that physical block. So even if they are combined, then it's still going to be located at the very tail end, but in some cases they are separate. Okay. So an interesting thing here, so now we are looking at TRIP-R, which is located upstream of the promoter region. And this is actually a sequence that codes for a repressor protein. So this is not something that's going to be transcribed down here. But this repressor protein, if it's allowed to bind to the operator, then that will act as a physical block, which will prevent transcription from occurring. Now, something that is interesting here is that this TRIP-R is going to be constitutively expressed. So this repressor protein is always going to be made. But we just said that the TRIP operon is a really good example of how like regulatory gene expression works, not constitutive. And the reason why that is because even though we always have this repressor protein being made and being present here, it's only going to function as a repressor if it binds to the operator sequence correctly. So if it's out here and not actually binding, then it's not really a repressor at that point because it's not actually doing anything. It's only when it can physically bind that it is able to repress transcription from occurring. That's just what I said. Okay, and so in the trip operon, we say that the repressor protein is born inactive. And so thinking about what that means, the default state of the repressor protein is to not bind to the DNA. So by default, even though these repressor proteins are being made, by default, they're not going to be binding. They're just going to be floating out here and allowing transcription to occur. That changes due to the presence of tryptophan. So this repressor protein is an allosteric protein. So hopefully the word allosteric reminds you of non-competitive inhibition of enzymes, which we talked about before. And we remember in that scenario, that was saying, oh, this is when another substrate binds elsewhere other than the active site on an enzyme, and it causes the active site to no longer be functional for whatever reaction the enzyme is used for. So similarly, the repressor protein, even though it has a binding site for the DNA, it's going to have a separate binding site elsewhere on the protein that will bind another ligand, which in this case is tryptophan. And when that ligand binds to the repressor protein, the conformation is going to change of the entire repressor protein in such a way that the shape of the DNA binding site is going to also change in a way that it either prevents or enhances the affinity for binding to the DNA. So in this case, TRP is acting as a co-repressor. And a co-repressor is what we call this allosteric binding that is improving the binding affinity of the repressor to the DNA. So we remember, okay, normally you're not able to bind, but once we add tryptophan, the shape of this protein is going to change and then it can bind, which will prevent these genes from being transcribed. And so the reason why this is important is because we talked about here that the cell doesn't really want to produce all these enzymes if they already have the tryptophan that they need. So they're able to kind of use tryptophan as this allosteric site to say that, hey, we already have enough of this tryptophan here, as you can see, because I have tryptophan bound to this repressor. So now, by binding to the DNA, I can say we don't actually want these proteins because they're not needed at this point. So what type of regulation is this, and how do we know? Well, we know that it's repression, also called negative regulation, because we know that like the default state is going to be transcribing is occurring. The reaction is occurring as normal until something happens that causes a change that stops or reduces the level of transcription. And so this change in the trip operon is the addition of tryptophan, which again modifies the shape of the repressor protein so that it is allowed to bind now. So overall, we can think of, okay, the normal transcription activity is going to be high, and it's going to be stopped by the addition of the new thing or the new condition. If this was positive regulation, we would expect to be the reverse, where we have low transcription that's then stimulated or activated by something. So you can kind of think about like, what's the net change? Is it going up or down in terms of transcription levels? And that will tell you if it's positive or negatively regulated. Okay, so now you can kind of test yourself. What do we expect to happen under low tryptophan conditions? It's kind of just a review of what I already said, but so when we have a low tryptophan condition, we're going to have a lot of this inactive repressor, which again is going to still be present. It's still being transcribed and expressed, but this shape is not able to bind to the operator. So the reaction is occurring as normal, and we're getting all these enzymes that will create tryptophan. However, when that tryptophan synthesis is building up, because all of this is allowing tryptophan to be made, once those levels get sufficiently high, what's going to happen is that the tryptophan will now be available to bind to an allosteric site of the repressor, which we can see it changes the shape. And now this shape is allowing the repressor to physically bind to the operator. And so again, this is the RNA polymerase, and it'll just run into this. It won't be able to actually go down the DNA at this point. So that's overall an example of the trip operon. The other classic example that we talk about is the lac operon. And this one is a little bit more complex. You'll see why in a moment. But it's also a really, really good way of testing your knowledge about gene regulation because it has that like an extra step of complexity to it. Okay, so the lac operon, again, is present in E. coli. And essentially E. coli always favors glucose as its primary energy source. So when glucose is present, it doesn't really matter if lactose is there or not. It's always going to preferentially choose glucose to break down into energy. However, if glucose is depleted for some reason or absent, then it's going to say, okay, we're going to use the lactose now. It's kind of our second choice, but we got to do it. And so when lactose is actually needed, the cell requires two particular enzymes in order for that to be broken down and to be actually like used as an energy source. The two enzymes are called beta-galactosidase and lactose permease, and I'll kind of go over those a little bit more in a second. But essentially, if you run out of glucose and you have lactose, those two enzymes are essentially required for this to be utilized. So how that relates to regulation is that we want the cell to know, okay, we have lactose present and we need it, so let's synthesize these two enzymes now. But if glucose is present, or if lactose is absent, there's no reason for these enzymes to be made. Okay, and the reason why lactose is always the second choice compared to glucose as an energy source can be kind of seen from this reaction here. So how lactose works as an energy source for E. coli is that it's broken down by this beta-galactosidase enzyme, which is one of the required enzymes that I was talking about, and it breaks this down into galactose and glucose. So we can see essentially all it is doing is it's just repleting the levels of glucose. So if we already have free glucose available to use, it's going to be faster and like energetically, it's going to just be easier to use the glucose directly rather than go through this extra step of synthesizing these enzymes and then doing this reaction when you're just creating the same product that you already have. So that's why you would only ever get this reaction happening if the glucose has been depleted. Okay. Oh, and also, so this beta-galactosidase is required to catalyze this reaction. The reason why lactose permease is required, which is the additional enzyme, remember we said earlier that two enzymes are required, that actually helps with the transport of the energy source like into the correct place. So if you don't have lactose permease, even if these products are being made, if they can't be transported to the right place, then they're not going to be able to be used as an energy source. So that's why both of those are operating under the same operon, because the same conditions will apply. Okay, so now we can look at these experimental results, and based on what we just talked about, we should hopefully be able to understand what's happening here. So we're looking here at these three petri dishes of E. coli and seeing, okay, is beta-galactosidase being produced? And remember, beta-galactosidase is required only when lactose needs to be broken down. So under this first petri dish, there's glucose only, no lactose. So as expected, there's no beta-galactosidase presence because glucose is always preferred and there's no lactose anyway. So there was really no reason for that to be produced. Under treatment number 2, we have a mixture of glucose and lactose. We're still not seeing any beta-galactosidase production because even though lactose is available for that to happen, glucose is always going to be preferentially preferred. So you're still not inducing this expression here. In treatment number 3, where we have lactose only, this is going to be the situation where now we are producing beta-galactosidase because we have a lack of glucose, which signifies, hey, we really need to synthesize lactose now. And the lactose is available. So it's kind of signifying we're allowed to do this, we have this. If we had an additional treatment petri dish that had no glucose or lactose, we would not see any beta-galactosidase because lactose isn't present for that to even occur. And that's just useful because I don't want you to just look for absence of glucose to determine whether or not this is being produced. Not only does glucose have to be absent, but lactose has to be present. So in other words, what that means is that here, lactose is acting as an inducer of the expression of beta-galactosidase. So we require lactose in order for this to be expressed. And glucose is simultaneously acting as an inhibitor of the expression of beta-galactosidase. So presence of glucose is going to prevent the reaction, whereas lactose is necessarily required for the reaction to occur. So the reason why the lac operon is so useful, but also a little bit more complex, is that you're having two different mechanisms of regulation, and they're working in opposite directions of each other. So this is just essentially saying what I just said, so you can look at this later. So previewing the lac operon structure, this is hopefully a familiar picture. We have the promoter, the operator sandwiched in between the promoter and the genes. And then these are the coding sequences for the two genes that are required, the beta-galactosidase and the lactose permease. And this is helpful because since they're regulated by the same thing, they'll either both be turned on or both be turned off, which is efficient because otherwise we'd have to activate them or inhibit them both separately. Okay, so now looking at how regulation actually occurs in the lac operon. So now we're kind of extending the view of this to see, okay, look, we have this coding sequence upstream of the promoter that codes for a repressor protein. Now this here should look really familiar to you because it's almost the exact same structure as the trip operon, where we also saw that there was a upstream coding sequence that coded for a repressor. And they're similar in some ways and different in other ways. So how they're similar, how this one is similar to the one that we saw in the trip operon is that the repressor coding sequence is always going to be, so this repressor protein is going to be always there. It's just going to depend on whether or not it can bind. That will determine whether or not it actually can repress the reaction or not. So that's how it's similar. The repressor protein is different than the trip operon in a very important way. So in the trip operon, remember that we said that the repressor protein was born inactive, which we remember means that the repressor by default cannot bind. So by default, the repressor protein is just floating away from the DNA and it's not actually repressing anything at that point until the tryptophan is added, in which case it binds to the repressor protein, which then changes shape, which favors binding of the DNA now. So now it's able to attach to the DNA and function as a repressor and prevent that reaction from occurring. So in this case, the ability to repress transcription depends on the presence of tryptophan. The presence of tryptophan is required. In the lac operon, it kind of works in the opposite direction. So the repressor protein is born active, which means that in the default state, the repressor is going to be bound to the operator. So by default, the transcription is going to be blocked in this case. Now what happens here is that when lactose is added, when we have lactose present, it's going to function in the same way as the tryptophan here, in that it's going to bind to an allosteric site and change the conformation of the repressor protein. But now, instead of enhancing binding, it's going to decrease the ability to bind to DNA. So now when we add lactose, it's going to take that repressor protein that's already bound to the DNA and make it fall off the DNA. So essentially, now that transcription can occur whenever we add lactose. So essentially, the ability to repress transcription depends on the absence of lactose. So we can see how these are kind of opposites of each other. This is just a little bit of a more graphic depiction of what's happening. So we can say, okay, naturally, in default state, we're going to have the repressor being bound, physical block of the operator, which prevents this RNA polymerase from binding. So naturally, this is going to be inhibited. However, when lactose is present, what's going to happen is, it kind of doesn't show this step, but the lactose is going to bind here to the actual bound DNA, or to the bound repressor. But then that's going to cause the repressor to change shape. So we can see, okay, it changes from this circular shape to square shape, just for the sake of this diagram. That's not actually what happens, but the shape is changing in a way that now it's no longer able to be bound at this location. So it's going to pop off, and that creates this extra space for the RNA polymerase to be effectively recruited, and transcription can occur. So that's why we say this is, the repressor is born active, because it's naturally going to be in this active state. Okay, so now let's recall these results that we were looking at earlier. So we just said here previously that when you add lactose, transcription's occurring, right? Then we look at here and we remember, oh, treatment 2 didn't show beta-galactosidase production, even though we had lactose. And why is that again? It's because the lac operon is also regulated by glucose. Remember, it has two forms of regulation here. So the regulation by glucose acts a little bit differently. So this is an even more detailed graphic of the lac operon. And we can see that upstream of this promoter region, we have this additional thing, which is called the CRP-CAMP binding site. And in this lac operon, this binding site is going to act as an activation site. So when this is bound by the correct substrate, it's going to be really happy and activate this, like highly upregulate the transcription that's happening here. And so the reason why this is related to glucose, presence or absence, I'll show you in just a second. So this will become more clear, like how this is actually related to the levels of glucose. But first, I just wanted to say, okay, so in this binding site, the thing that's essential in order for this to be activated is the CAMP molecule. So the CAMP and CRP are going to bind together and form a complex, which forms this shape that can fit into this active site, or to the activation site. And since it's bound, it's really happy and will upregulate this transcription a lot. However, when CAMP is absent, even if CRP is still here, it's not going to be it's not going to be in that favorable shape that will allow that binding. So the absence of CAMP is going to prevent this activation site from being bound, which means that the transcription level is going to be relatively low. So now going into, okay, why is the presence of CAMP a proxy for absence of glucose? So we can look at here what reaction is happening. So in this reaction, we have this ATP molecule that when there's low levels of glucose, when there's no glucose around, an enzyme called adenylyl cyclase is going to break down this ATP into CAMP and 2-phosphate groups. And we remember CAMP was required for that activation site to be bound and for that upregulation to occur. So when we have low levels of glucose, this will be produced and this will be upregulated. However, glucose actually is an inhibitor of this enzyme, adenylyl cyclase. So when you have a high level of glucose, you are knocking out the function of this. So anything that's like past this arrow cannot be produced. So you're not going to get any CAMP production, and you're instead just going to have this reservoir of ATP. And what that means is that you're going to have this situation where we still have CRP, but that doesn't matter because we can't activate that correct conformation with the CAMP. So essentially, that's kind of how we use CAMP as like a proxy of glucose, is because those things are inversely related. Okay, so now we can think, is the lac operon an example of positive or negative regulation? And first, I wanted to review these terms again. So I know I already covered this, but the terms are like super similar to each other and people get confused between them. So just to get you guys used to the terms again, we can remember that positive regulation, which is the same thing as induction, is essentially any method that will result in an increase in the amount of transcription from its natural state. So the two primary ways that this can happen is by inactivating the repressor protein. And this would be called an inducer. So if you think about, okay, if we're inactivating something that represses a reaction, that's the same thing as saying you're inducing that reaction. So that's why this is called an inducer. Another way that we can increase transcription is by activating that activation site. So if we were to activate CAMP production, then we're going to promote the binding and blah, blah, blah. We talked about that already. So those are going to be examples of positive regulation. Negative regulation is also going to be called repression, which is any method that results in a decrease in the amount of transcription like from its natural state. So this can be accomplished by activating a repressor protein. So in this case, this is called a co-repressor, or you can inhibit the activator. So by preventing the activation site from being bound, then you're not going to get that upregulation. So the important thing to note here, I think, is that the inducers and the co-repressors are doing functionally the exact same thing. They're both going to be binding to an allosteric site of the repressor protein and causing some sort of conformational change. The only reason why they have these two different names is because it's signifying what effect it's having on the binding affinity of the protein to the DNA. So if we think about an inducer, you are essentially making the binding worse, so the repressor protein will fall off. Whereas if you are activating the repressor protein, you're increasing the binding affinity for the DNA, which allows the repressor to actually repress the reaction. So they're doing the same thing, but they have different outcomes because of that conformational change. Okay, hopefully that makes sense. So now we can kind of think back, okay, what is the lac operon doing again? Hopefully you can tell me both. So there's positive and negative regulation occurring here. So it's called dually regulated. So we can look at positive regulation. There's two main mechanisms. So for inactivating the repressor protein, we can hopefully recall that lactose was acting as an inducer, because in its natural state, the repressor is going to be bound and it's going to be inhibiting the reaction. But when we add that lactose, it's changing the shape so that the repressor protein falls off and suddenly the transcription can occur. So this is positive regulation because you're increasing the net availability of that transcription to occur. Additionally, we regulate by activating the activation site, which we do by the CAMP production, which we remember, okay, when we have a high level of CAMP, that's going to be a positive regulation because you're going to be exciting the ability to transcribe. And we can recall that this is related to the glucose concentration, which is why we're able to say that it's regulated by lactose and glucose, rather than just like lactose and CAMP. So negative regulation is occurring from the default state of the repressor protein, where the repressor is bound and it's blocking the transcription levels. But it also can inhibit the activator. And that will occur when there's a high level of glucose. So you can recall that glucose will deactivate or denature the adenylyl cyclase enzyme, which is what produces CAMP. So if you have glucose, you're not going to have CAMP, which will essentially prevent the activator from working, and that reaction isn't going to happen. So now we can use all this information together to kind of predict what will happen in these situations. So here we can look at this top one. We have a wild type repressor protein sequence, which essentially wild type here just means that it's normal, it's functioning as it should. And we have no lactose present. And that's all the information that we're given. So we should be able to use that information to say, okay, is there going to be high or low transcription? And try to think about it before you like listen to the next slide. But we can tell automatically, just given this, that there's going to be low transcription. Because if there's no lactose present, the repressor protein is going to automatically be bound, right? It's going to require that lactose to inactivate it and make it fall off. So if it's not there, then it's always going to be bound. So regardless of what is happening with the glucose, you're always going to get this low level of transcription. Okay, so now we can look at this next one, where we have the wild type repressor sequence again. So again, it's just functioning normally. But now we have the presence of lactose. And so here, you might be tempted to say, oh, so we know that it must be high because now we're able to get that repressor protein off. And that's tempting to say, but you have to remember that this operon is dually regulated. So here, we know that technically lactose transcription could occur if it was only regulated by lactose, but we have to also take into account glucose. Because if you have high levels of glucose, you're going to be not activating the activation sites, which is going to be what propels the reaction forward. So if you have high levels of glucose, you are still not going to get this transcription occurring. And we can kind of remind ourselves of that based on these experimental results. So that is kind of symbolic of this second Petri dish, which is when we have glucose and lactose. So even when we have lactose present, it's still not going to be synthesizing the enzymes because glucose is the preferential source, which is what we saw here. We always want it to be glucose first and then lactose only if it's needed. Okay, so hopefully that makes sense about why you can't say this is automatically going to be high just based on this. Okay, what about this result here? Here we have a mutant repressor protein coding sequence, which we can assume that that mutation is going to cause the protein to no longer be able to be expressed. So this mutation is going to prevent any repressor protein from being made. And now we can try to predict, okay, how would that affect how this is regulated? And try to think about it before I start explaining, so you can pause and try to think what will happen. But essentially, when we lose that functionality of the repressor protein, now it doesn't matter whether or not lactose is there, because whether it's there or not, there's no repressor protein available for it to change shape of. So normally, if you had the wild type, the repressor protein would be bound, but then if lactose comes in, then it would be unbound. But here, there's nothing repressing to begin with from this repressor protein. So lactose is no longer a regulator of the lac operon in this case. However, we still have this result of we can't say definitively because we need to know what's happening with glucose. So here, even though we kind of made this lactose negligible, it doesn't really matter if it's there or not, you still have to say, oh, okay, is glucose high or low? So if it's going to be high, remember that it's going to inhibit the CAMP, not bind to the activator, and this will be low. If the glucose level is low, we're going to have that increase in synthesis of the CAMP, which can bind to the activator site and then allow this transcription to occur. Cool, so that's essentially all that I had to go over concept-wise. I just wanted to say real quick a little bit of advice that I have. I think that the main reason that students find this topic to be confusing isn't because it's hard conceptually, it's just because of the terminology and how similar all these terms are and how they're all like kind of opposite of each other or like related to each other in like a really close way, but like have different meanings. So my suggestion for dealing with this is instead of trying to remember, okay, this is what happens when you have an inducer, or this is what happens when you have an activator, or like something like that, try to focus instead on like the literal meanings of those words and what that tells you about what's happening in the reaction. And the reason why is like, so for an example, like on an exam, you could have a question that's phrasing something like, you're inactivating a repressor. And it would be up to you to know that, okay, inactivating a repressor is functionally the same thing as saying that you're inducing transcription. So even though it doesn't say this explicitly, you have to know that these two concepts are related and why. And hopefully it should be intuitive. But if you just focus on memorizing rather than understanding those underlying concepts, it can be really easy to like mix these things up. So yeah, that's my first big piece of advice. And also, second piece of advice is just to read the questions really, really carefully. Because, for example, if a question about what will happen if you inhibit an activator, the answer is going to be totally different if you interpret it as being inhibiting a repressor, for example. So like just taking that little bit of extra time to highlight, okay, what is it asking? What direction is it asking? And what thing is it asking about? Is it happening to an activator or repressor? Like just really taking that extra time because I would really hate to see people lose points for concepts that they know because of just like reading too fast or not being careful. Okay, cool. So that's all that I wanted to talk about. If there's any questions, please let me know. I'm happy to go over this with you. I tried to, again, leave the prediction slides empty so that you can kind of go through like here and predict on your own before looking at the answer slides. So take some time right before the exam to kind of go back through these and make sure that you're getting the correct answers, and that will tell you what you need to focus more on in terms of studying or not. So cool. Thank you guys. Bye.